So here is DNA replication. So this is the DNA strand that we were given. So this is the original strand. So just doing this, we are going to see the complement base pairing, which is going to be the opposite of this. But since Chargaff's rules, we see that T goes with A, which is thymine and adenine, and then cytosine and guanine go together. So we will actually see A, T, G, C, C. And that is the complement base strand. So in this, before we start at the origin of the DNA, basically this is all going to be together and we wouldn't see this kind of break. But since we see this, helicase, we'll just draw a circle right here. Helicase is actually what kind of unzips or unwinds the actual protein strands, and this is an enzyme. As well, we see here to refrain from these two strands that were being broken apart, actually refraining to come back together. This is going to be represented by the SSB proteins, which are single-stranded bonded proteins. We also see what is called topoisomerase, which basically what he does is he just avoids all this is supercoil. So if we had supercoiling for replication to carry out properly, we need to keep them separated. So what he kind of does is he refrains the rest of the strand to overcoil, which can also cause issues later on with the polymerase and the primers. Now we actually see primase come in, which basically places the primers on the leading and lagging strand. Now this is the leading, going 5' to 3', and this is the lagging 3' to 5'. So we do see primase, let's label that, which actually lays down a primer. And we're just going to label that pre. So with these primers at each end of the leading and lagging strand, we will actually see DNA polymerase, which starts at each end of the primers. So let's just draw a little circle right here. So what DNA polymerase actually does is it kind of builds the new strands, which occurs at the 5' to 3 direction. We eventually will see bases being placed from DNA polymerase. But let's just address the 3' to 5'. This is the original strand as well as the 5' to 3'. These are complementary of each other. That's why we see this. So basically we are seeing this is at the top and then that complementary strand will be at the bottom. But we do need them to be complementary of each other. And that is why there's the 3 to 5 at the top, the 5 to 3 at the bottom. And again, these are the original strands. So back to DNA polymerase, we do see them building new strands, which is right here. We do see them building in the 5 to 3 direction. That's why you see the 5 to 3, the 5 to 3, and that is complementary to each other, that 5 to 3', and then that 3 to 5' right there. So now as we see it start to unwind more, and then we'll just bring that together. This is still 5' and this is still 3'. And helicase is still making this all happen and unwind. But where DNA polymerase comes in is building and constantly building these strands. Now this top strand right here, going from the 5 to 3 direction, this is the leading strand. Because as it leads, the unwinding process, it keeps on building at the top. Now down here, we still see the same thing, but this happens much slower, and this is why this is called the lagging strand. So why this is called the lagging strand is every time this is continuously going, because it's the 5 to 3' direction, the DNA polymerase lags, and every time a primer has to be put down, it has to build. But before it DNA polymerase to even start building the new strands, it has to place a primer down. So after this is happening, we kind of see these little fragments, and these are called Okazaki fragments. So since these primers are actually made of RNA, we do need to replace them with ligase. Now, DNA ligase actually is kind of the gluing agent to this. So right here you see these gaps. So right here, we would see ligase come in and actually glue these strands together, or these fragments together. And that, again, is ligase. So at the end of replication, we end up having 2 identical DNA strands. And this all came from this one original DNA strand. Now, this process here is actually called semi-conservative, due to this being the new strand. And what that means is you can see here the green lines, or the green strands, are the new strands. But the red ones were actually the original strands from this original DNA helix, double helix. And this is how DNA replication is done.